What's up, biology students? It's me again. In our last video, we talked about meiosis and how this process contributes to sexual reproduction by forming the cells necessary for an organism to engage in sexual reproduction. These specialized cells, called gametes, allow chromosomes from separate organisms to combine in the formation of a new organism, and this is how genetic traits are passed from one generation of organisms to the next. In today's video, we're going to learn more about how the passing of chromosomes from one generation to the next influences the traits possessed by that next generation. The chromosome theory of inheritance tells us that genes are carried on chromosomes, and that offspring inherit their chromosomes, their genes, and therefore their traits from their parents. Humans, and a lot of other organisms, have chromosomes that come in sets of two, called homologous pairs, because each chromosome in a homologous pair has the same set of genes as the other chromosome. Because of the way chromosomes are divided up during meiosis, offspring inherit one chromosome from each homologous pair from their female parent, and one chromosome from each homologous pair from their male parent. This works out because the gametes produced by meiosis are haploid, each containing a half set of chromosomes. Gametes contain no homologous pairs, but rather contain one chromosome from each homologous pair. When gametes combine during sexual reproduction, the result is a diploid cell with homologous pairs of chromosomes. Because our chromosomes come in pairs, that means that we have two copies of every gene. But because we inherit each copy from a different parent, we may or may not have the same allele for that gene. Alleles are the specific form of a gene. For example, we have a gene for eye color, and for that gene, we could have an allele for blue eyes or an allele for brown eyes. These letters represent the different forms of a gene, and usually we use one letter per gene that represents the specific allele by making that letter either capitalized or lowercase to represent its specific form. If an individual is heterozygous, that means the individual has two different alleles for the gene. In this case, it means our individual has one allele for blue eyes and one allele for brown eyes. That's our big B, little b individual here. If an individual is homozygous, that means the individual has two of the same allele for that gene. This could mean that our individual has two alleles for brown eyes, our big B individual here, or has two alleles for blue eyes, our little b individual here. These terms, heterozygous and homozygous, describe an organism's genotype, or its genetic makeup. An organism's genotype determines the organism's phenotype, or its physical appearance or innate behaviors. For example, depending on which alleles an individual has, it could have a big B, little b genotype, which is called heterozygous, a big B genotype, which is homozygous, or a little b genotype, which is also homozygous. Based on the combination of alleles possessed by our individual, this person might have blue eyes or brown eyes, and this would be our individual's phenotype. Here's how it works. For a lot of genes, there is a dominant allele and a recessive allele. Dominant alleles are called dominant because you only need one copy of this allele to have that phenotype. So both of these individuals would have brown eyes because brown is the dominant allele, and both of these individuals have at least one brown allele for this gene. Recessive alleles, on the other hand, are called recessive because you need two copies of this allele to have that phenotype, and the presence of a single recessive allele may be hidden by the presence of the dominant allele that ultimately gives the individual its phenotype. Most of our traits are determined by several genes, not just one. Eye color, for example, is determined by at least two genes. One of these genes, the HERK2 gene, has two alleles, brown and brown is dominant over blue. The other gene, the G gene, also has two alleles, green and green is dominant over blue. This chart shows us which combinations result in brown, green, or blue-eyed phenotypes. As you can see, more combinations result in the brown phenotype than any other color, but both the HERK2 gene and the G gene influence the phenotype of the individual. These patterns of inheritance were originally discovered by an Austrian monk named Gregor Mendel. Gregor Mendel was living in a monastery in the mid-1800s, devoting his life to religious and scientific study. Mendel was studying pea plants, looking at how a variety of traits were passed from parent plants to the offspring produced from seeds. It is important to note that at this point in history, we had absolutely no knowledge of DNA or genes, and we had really only just begun to learn about the basic structure and function of cells. But because we had been growing plants and raising livestock for a long time by then, we had a pretty good sense that offspring resembled their parents in terms of their physical appearance and behavior, and had a pretty good idea that traits were somehow being passed from parents to their offspring. By carefully controlling which pea plants were allowed to breed, and by making careful observations about the offspring grown from seeds produced by crossbreeding, Gregor Mendel discovered that there are predictable patterns to how traits are passed from parents to their offspring, and that there was more to this inheritance than a simple blending of parents' traits. He looked at whether the plants produced plump versus wrinkled seed pods, had round versus wrinkled peas, had green versus yellow peas, and green versus yellow pods. He observed whether the plant had purple or white flowers, and whether they grew up to be tall or short when grown under the same conditions. The height that a plant is capable of growing to, for example, is determined by a single gene. This gene has two possible alleles, the tall allele, or big T, and the short allele, or little t. That means that, for this gene, our pea plant could have one of three possible genotypes: homozygous dominant, big T, homozygous recessive, little t, or heterozygous, which is big T, little t. Depending on which genotype a pea plant has, it could grow to be tall or short. Because the tall allele is dominant, both the homozygous dominant and the heterozygous genotypes will allow the plant to grow tall, but the homozygous recessive genotype will limit the height of the plant and will keep it short. It's important to note that plants have a life cycle that is very different than ours. They do something called alternation of generations and exist for part of their lives as a diploid sporophyte and for part of their lives as a haploid gametophyte. That isn't really reflected here, and it's not really the focus of our video, but it is important to mention so that we don't go around thinking that plants and animals do everything in exactly the same way. With that disclaimer behind us, suppose our pea plant is heterozygous. That means that each cell contains one tall allele and one short allele. When this plant makes cells by way of meiosis, these cells will either contain one tall allele or one short allele, but not both. Of the four possible gametes this plant could make, 2 would contain the tall allele, and 2 would contain the short allele, in 50-50 proportions. If our plant is homozygous for the tall allele, then all the gametes produced by that plant will contain the tall allele because it's the only allele that this plant could possibly pass on. Likewise, if our plant is homozygous for the short allele, then all its gametes will contain the short allele. Suppose that we, like Mendel, were to breed or cross a tall plant with another tall plant. What is the probability that the seeds produced by this cross will produce offspring that are also tall? The answer to that question will depend on the specific genotype of each parent. But let's take a second to talk about probability. Probability describes the odds or chances of a particular event happening. For example, if you flip a coin, there's a 50% probability that it will land on heads, and a 50% probability that it will land on tails. Does this mean that if we flip a coin twice, it will land on heads exactly once and on tails exactly once? Not at all, because each flip of the coin is a separate event with its own probable outcomes. Inheritance of genetic traits works in much the same way, especially if you are only observing one trait controlled by one gene. When we start to consider multiple traits controlled by multiple sets of genes, things start to become a bit more complicated. So how do we figure out probability? Let's look at an example. Suppose we're rolling a pair of dice. What's the probability of rolling a combined total of 7 on these two dice? Well, we know that there are only so many outcomes for each individual die. On the first roll, we have an equal chance of rolling any number 1 through 6, and we have a 1 in 6 chance of rolling any one particular number. The same is true of our second roll. Since there are 6 possible outcomes for each die, that means a total of 36 possible outcomes when both dice are rolled together. Of those 36 total possible outcomes, 6 combinations add up to 7. 6 out of 36 is the same as 1 in 6, so we've got a 1 in 6 chance, or a 16.7% chance, of rolling a 7 on 2 dice. This chart I've created here is called a Punnett square, and it's a tool for helping us to determine probability. You'll see a lot of Punnett squares when the subject is inheritance of genetic traits. But let's get back to our question. Suppose that we were to breed or cross a tall plant with another tall plant. What is the probability that the seeds produced by this cross will produce offspring that are also tall? Let's suppose that each plant is heterozygous, meaning that the cells of each plant possess one dominant tall allele and one recessive short allele. With these alleles, that means that the gametes produced by these plants could either contain a single dominant tall allele or a single recessive short allele, but not both. To set up our Punnett square, we write the possible alleles from one parent along the top and the possible alleles from the other parent along the side. In this grid, we can see all the possible combinations of these alleles. One possible combination results in seeds that are homozygous dominant, containing two tall alleles. One possible combination results in seeds that are homozygous recessive, containing two short alleles. Two possible combinations result in seeds that are heterozygous, containing one dominant tall allele and one recessive short allele. Since 3 out of the 4 possible combinations have at least one dominant tall allele, that means that there is a 75% chance that any given seed produced by this cross will grow into a tall individual, and a 25% chance that a seed will grow into a short individual. Likewise, if we planted 100 seeds produced by 100 crosses, we might predict that roughly 75 of them will grow up to be tall, and roughly 25 of them will grow up to be short. Our predictions are less likely to be accurate if we consider only a few seeds, in much the same way that it is possible to flip a coin and have it land on tails 5 times in a row. But if we flip that coin 100 times, and if we plant 100 seeds from 100 different crosses, our results are far more likely to match our predictions based on the laws of probability. If both plants are homozygous, then both plants have only tall alleles to pass on to the next generation, and the only possible combination based on these gametes is another homozygous offspring with two tall alleles. So if both parents are homozygous for the tall allele, there is a 100% chance, a 100% probability, that their offspring will also be tall. That's not terribly surprising, but our Punnett square here shows us why this is the only possible outcome. If only one of our parent plants is homozygous for the tall allele, and the other is heterozygous, we still have a 100% chance that the seeds will produce tall offspring. But there is also a 50% chance that a seed will be heterozygous and will contain at least one short allele that could be passed on to its offspring. Now suppose that we cross a homozygous dominant tall plant with a homozygous recessive short plant. The tall plant can only pass on tall alleles because those are the only alleles that it has. And the short offspring can only pass on short alleles because those are the only alleles that it has. And that means that every seed produced by this cross will have one tall allele and one short allele. They will all be tall, but they will also all be heterozygous and may pass on either a tall or a short allele to their offspring in the next generation. If the homozygous dominant tall plant in this scenario is replaced with a heterozygous tall plant, then that means a 50% chance of producing a tall offspring and a 50% chance of producing a short offspring, because 2 of the 4 possible combinations have at least one tall allele, while the other 2 possible combinations contain only short alleles. And finally, if we cross 2 short plants, then the seeds produced by this cross will all produce short offspring, because short alleles are the only alleles that can possibly be passed on by either parent plant. And once again, our Punnett square tells us why this is so. But suppose we are interested in looking at more than one trait. Well, the more traits we look at, the more possible combinations there are, which is why no 2 people on the planet are completely alike. To illustrate this diversity, let's take a look at an example involving 2 traits, seed shape and seed color. Each of these traits has 2 possible alleles. In terms of shape, round seed alleles are dominant over wrinkled seed alleles, and in terms of color, yellow seed alleles are dominant over green seed alleles. To understand the patterns of inheritance when dealing with more than one trait, we need to understand the laws of segregation and independent assortment. In this simplified diagram, A1 and A2 are homologous chromosomes and contain the same genes. B1 and B2 are also homologous chromosomes and also contain the same genes, but the B chromosomes contain different genes than the A chromosomes. During meiosis, chromosome A1 is segregated or kept separate from chromosome A2. Likewise, chromosome B1 is segregated or kept separate from chromosome B2. You'll never produce a cell that has both the A1 and the A2 chromosome if everything goes as it should. However, during meiosis, A1 is sorted independently from B1, and A2 is sorted independently of B2, meaning that the 1s may or may not be sorted into the same cell, and the 2s may or may not be sorted into the same cell. Because homologous chromosomes are segregated from one another, and because non-homologous chromosomes are sorted independently, that means that if we are interested in two traits carried by two different chromosomes, then there are 4 possible combinations of alleles that could make it into the gametes produced by that organism. We can see those 4 possible combinations here in this figure, and each combination is as likely or as probable as the others. That means that if we look at one gamete produced by this organism, there is a 25% chance that this gamete will contain combination 1, a 25% chance that it will contain combination 2, and so forth. Similarly, that means that if this organism were to produce 100 gametes, roughly 25 of them would contain combination 3, while 25 of them would contain combination 4, and so forth. And remember, the more traits we are interested in, the more possible combinations there will be. In this figure, we're looking at only the possible combinations of 2 chromosomes. 3 chromosomes could combine in 9 different ways, 4 chromosomes in 16 different ways. And for all of our 23 sets of chromosomes, there are over 500 different combinations. And that's not even considering the fact that genetic recombination during crossing over mixes things up even further. Let's take a look at how this applies to Mendels pea plants. Suppose we cross a plant that grows round, yellow seeds with a plant that grows green, wrinkled seeds. Let's further suppose that both parent plants are homozygous for each trait. This plant has 2 round, or big R alleles, and 2 yellow, or big Y alleles, and this plant has 2 wrinkled, or little r alleles, and 2 green, or little y alleles. Their genotypes are big R, big Y, and little r, little y, respectively. Because of the law of segregation, each gamete must contain 1 allele for each trait, either the black R or the red R, and either the green Y or the orange Y. Furthermore, because of the law of independent assortment, each of these alleles for the first trait could be paired up with either of the alleles for the other trait, black with green or black with orange, red with green or red with orange. The same is true of the other parent plant as well. In this example, because both the parent plants are homozygous for both traits, the gametes produced by this plant will all be the same, and the gametes produced by this plant will also all be the same. However, the color coding here shows us that there are still 4 possible combinations for each parent plant, 4 possible configurations for the gametes that each plant makes. If we make another Punnett square to see all the different ways these gametes could combine during sexual reproduction, we find that all the possible combinations would result in offspring that are heterozygous for both seed shape and seed color. So far, these results are not terribly exciting, but suppose we take 2 of these heterozygous offspring and cross them to make more seeds. Well, now here's where things get a little bit more interesting. This time, we're crossing 2 plants that both produce round yellow seeds, and each of these parent plants has the genotype big R, big Y, because each of these plants has 1 round allele and 1 wrinkled allele, as well as 1 yellow allele and 1 green allele in each cell. Just like before, there are 4 possible combinations of these alleles that could end up in the gametes produced by this plant. Those gametes could contain either 1 big R and 1 big Y, 1 big R and 1 little y, 1 little r and 1 big Y, or 1 little r and 1 little y. The same is true for our second parent plant. This time, if we set up our Punnett square with one parent's possible gametes along the top, and the other parent's possible gametes along the side, we see that there are still 16 possible combinations, but unlike last time, these possible combinations do not all result in the same phenotypic outcome. Our offspring look pretty different than one another. Of these 16 possible combinations, 75% of these, or 3 in 4, will result in offspring that also grow yellow seeds, while only 25%, or 1 in 4, will result in green seeds. Likewise, 75% of these combinations will result in round seeds, while only 25% will result in wrinkled seeds. Those numbers, 75% to 25%, or 3 to 1, are the same as we found when we were dealing with only one trait. But so far in this example, we're still really only dealing with one trait at a time. If we look at the combinations of traits, both for seed color and seed shape, we find that 9 out of the 16 combinations result in plants that grow round yellow seeds, meaning a 56% probability that a seed produced by this cross will grow into a plant that also has round yellow seeds. And statistically, this is the most likely outcome of this cross. 3 out of the 16 combinations result in plants with round green seeds, and 3 out of the 16 combinations result in plants with wrinkled yellow seeds, meaning a 19% chance that a seed produced by this cross will grow into a green round seed, and a 19% chance that a seed produced by this cross will grow into a wrinkled yellow seed. The least likely outcome of this cross, which would be produced by only one combination out of the 16 possible combinations, is a plant that grows wrinkled green seeds, and there is only a 6% chance that a seed produced by this cross will grow into a plant with wrinkled green seeds. That's because this phenotype requires 2 recessive alleles for each trait, and all 15 of the other possible combinations in our Punnett square have at least one dominant allele either for seed shape or for seed color, and oftentimes for both. Remember, Mendel couldn't see these alleles that we are depicting here, and he had no idea that these things that we now call alleles are versions of genes carried on these things that we now call chromosomes. All he knew about was what we now think of as the phenotype of each plant. But by keeping careful track of the characteristics of the offspring grown from seeds produced by each of his crosses, Mendel was able to propose a mechanism for what he saw, and this mechanism, which assumes that traits are controlled by a pair of alleles, has helped us to make successful predictions about how traits are passed from one generation to the next. This was Mendel's most important discovery, that there are predictable patterns to the way that traits are passed from one generation of organisms to the next. And this pattern could be explained if we assume that each trait is controlled by a combination of 2 alleles. Today, we know that the process of meiosis controls how these pairs of alleles in a single diploid cell are segregated and independently assorted into 4 haploid cells. And we have a better understanding of these cellular functions and processes that influenced Mendel's results. But Mendel's work laid the foundation for what we now understand about genetic inheritance today, and is the cornerstone of what we now call classical genetics. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand the basic patterns of genetic inheritance that we have discussed in this video, and why those patterns exist.